Hello, guys. I think let's get going. Uh,
Yes, sir.
what we'll do today is I looked around some site wherein we have questions for the non-coding RNA and related stuff. Maybe we can just go through that, and if someone has answer, we can see the answer. Okay, so the first one is mostly it's all present in the web, but I thought it's relevant. So the first question, people can read this. Is it
Yes, sir.
okay? Size is
Yeah,
good
yeah, we
enough.
can see. Yeah, yeah.
Okay,
Sir,
I'll
or you could
zoom.
send us the links to these questions and we can open it on our browsers.
Yeah. So maybe yeah, I'll do that. Okay, where is that? Yeah, this is the first one. We have three such kind. So mm, then the third one. Okay, it's pretty much we have covered, but we can just discuss and go ahead. Okay. So the first one, if you people can see it. Okay. So the first question is, yeah, mRNA of which of the following organisms does not undergo processing? So this one is very simple. Anyone can make out. Correct?
Bacteria. Yeah.
Yeah, bacteria. That's correct. Then rRNA, tRNA of prokaryotes does not undergo any post-transcriptional processing. Is it true or false?
It's false.
Prokaryotes, so it will be.
Sir, it's false.
Oh,
It's false. So it's false because there are a lot of endonucleases which makes it in the maturation of the rRNA or tRNA. Just like we discussed the other day, the circular RNA, in the case of archaea, you can think about it. Circular RNA, that there was maturation of some of the rRNA. Even in E. coli also, it is kind of process with endonucleases from the primary transcript to the mature tRNA or rRNA. And there is a lot of epigenetic modifications we discussed earlier also. Yeah. Then next is splicing of mRNA occurs after they arrive the cytoplasm. This is pretty much simple, but anyone can. True, false?
False.
False. So generally it happens in the nucleus, and without that proper processing, we know that mRNA or many of the other RNAs are not doesn't go exported to the cytoplasm. Then which of the following is not a type of RNA processing? Polyadenylation, capping, removal of exons, splicing.
C.
Removal of exons.
Removal of exons. So we don't do any removal of exons. Then elongation of transcription and RNA processing are all interconnected.
False, right?
It's false, correct? Elongation of transcription, RNA processing are all interconnected. Let's see. B. Okay, so it's not true, correct? Because there is answer also. There is a good thing about this website that answer is also there. Okay,
sir,
transcription,
question.
processing. Yeah.
You mentioned that removal of exons is not exactly a part of RNA processing.
Yeah, as such, there is no removal of exons, no,
It's removal
in
of
RNA processing.
Yeah, but in circular RNAs, you sometimes lose exons, right?
Yeah, but it's not. It's part of a splicing, correct? Generally, it's removed through splicing. In the splicing, that one, it might be an exonic region in one transcript. It may be a part of intronic, correct? So
Yeah.
there is no actual exon removal. So that is
Okay.
false.
Okay. Okay.
Yeah. Then next, the first RNA processing event is capping,
Excuse me, sir.
tailing. Yes. Yeah.
Sir, in the previous question,
Hmm.
in one of the previous classes, we saw that sometimes when transcription is going on, these non-coding RNAs can bind and they can affect uh, how the RNA is processed. So would that not come under this case?
Elongation of transcription and RNA processing are all interconnected. Is it all interconnected? No, no. Elongation is independent of RNA processing. Why do you think it is interconnected?
No, we saw that in some cases, some non-coding RNA can come and bind and interfere with transcription as it's getting elongated. So,
But that is regulation. It's not the processing of the RNA as such. No? It's coming
okay,
and then binding and then regulating the elongation rate. But as such, it's not RNA processing, I guess. Yeah.
okay, okay.
Hmm. Then the first RNA processing event is, this one is simple. Which one is this? Capping,
Capping.
tailing, splicing, editing.
Capping.
Capping. No? Capping is done by the addition of methylated ATGC.
Methylated G, right?
G. Yeah, methylated G, na? M7 G cap. Okay.
Yeah.
Then next, capping occurs after the mRNA synthesis is complete. Is it true?
Sir,
False.
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 It can
Yeah.
start before that, no?
Yeah, it generally starts before that. As the polymerase reaches the end of RNA, which of the following event does not occur as a response? Transport of polyadenylation enzymes, cleavage of RNA, addition of poly A at the 3' end, termination of transcription. Which one is it?
Addition of poly because it says
As the
as the
polymerase reaches the
polymerase
end of
reaches.
RNA, which of the following event does not
The adenylation
occur as a
polyadenylation enzyme will
response?
come, but it can't add poly A because polymerization has not ended yet.
Hmm.
Yeah, true.
Yes. Yeah. C. C is the addition of poly A. Correct. Uh, is that your answer? You said.
Yes, that's what he said,
You
sir.
can't add it
Hmm.
if the RNA
Hmm.
has not detached yet.
Yeah. Okay. Then about how many A are added to the nascent RNA in the 5' end during polyadenylation?
0. It's added in the 3
Nascent RNA
di.
in the 5' end during poly is 0. Yeah, correct.
Yeah.
Because it is 5' end.
Yeah, yeah.
The long tail of A's is unique to the RNA synthesized by RNA polymerase. Which RNA polymerase?
Two, I think.
2.
Yeah,
Polymerase 2.
yeah, RNA polymerase 2. Yeah. Then let's go to the next page. Up, if you see the next here, click there. Let's go. That will be more of non-coding RNAs. Okay. So non-coding sequence in mRNA is known as, this one is very simple, correct?
Introns.
Introns. Which of the following organisms have monocistronic DNA? Monocistronic
Bacteria.
DNA. Bacteria. Okay, correct. Then next, which of the following organism have overlapping genes? Overlapping genes. So
Bacteria only, right?
Virus, I guess.
bacteria. Virus is very common, but I think bacteria also has overlapping genes in some of uh, the locus is overlapping, but the answer, the more often is the
Sir,
viruses have a lot of overlapping
Uh,
genes.
Sir,
So because of the
sir.
small size,
Yeah.
Regarding the monocistronic, isn't it eukaryotes that have monocistronic? Not bacteria have
what?
polycistronic. Bacteria have polycistronic genes, if I remember right. Could you check?
Have monocistronic DNA. Monocistron.
Monocistronic means only exons,
Yeah,
right?
only cistrons.
That is
It's only
bacteria, no?
bacteria.
Yeah.
This means the genes are not disrupted by the presence of introns. Okay. B, bacteria.
Okay. All right. All right. All right. Okay.
So next one is no need to look so seriously, but just for information, it's 1977. First time introns was discovered when people were looking at adenoviruses. Okay, so this one is just kind of memory based, just to know, and there is nothing much. Uh, then let's forget this 5 and 6 because that is more of memory based. The amount of exon in the eukaryotic genome is higher than that of the introns. Is it correct or false? True or false?
False,
False.
false, false.
Yeah. So generally introns are much bigger than the exons. Eukaryotic, the mRNA, this one is kind of general knowledge. Many times people ask the mRNA of which eukaryotic protein lacks introns, hemoglobin, myoglobin, histone, polymerase. Any idea? Anyone? So
No, sir.
generally is the histones. Histone genes are very much well known not to have introns in their genes. Okay, so histones are the answer. Introns are nothing
What
more
is the reason, sir?
that we don't know why it is so, that we don't know. Histones,
Okay.
but it is observed in most of in high eukaryotes. Most of the time, it doesn't have introns. Histones maybe uh, because introns are supposedly introduced for better regulation. Maybe all these histones are very, very central to it for the organization of genome. Maybe it is so important that it doesn't tolerate any that kind of evolutionary changes in the gene. Maybe
Sir, and
that is.
also are histones present? Do we have the same histones in eukaryotes and prokaryotes?
No, in prokaryotes, we don't have the eukaryotic histones, but they do have DNA binding proteins which help in the organization of the DNA. And it is there.
Okay.
Functionally equivalent is there, but the protein exact protein equivalent is not there, histones.
Okay. Okay.
Then introns, this is again very straightforward. Introns are nothing more than genetic load in the eukaryotic genome. Is it true or false?
False.
False, false.
False. Correct. So many of the introns are regulated. Uh, it contains a lot of regulatory elements like microRNA targets, or
Yeah.
it regulates in the kind of exons to be included or excluded during splicing. It has its functional roles. Presence of introns facilitate the formation of several different mRNAs, thus increasing protein yield of different types, but from same gene. True or false?
So true.
True, correct. Yeah. So alternate splicing allows different proteins to be synthesized from the same gene, correct? So increases the number of different types of proteins are allowed to express. Yeah. So this is one side. Then next, let's go to the second side. Okay. It's about non-coding RNAs. Okay. So this site, after some time, after 7, 8 slides, it will tell you to register, but don't register, just refresh the page, then we can go ahead. That's what I figure out. Okay. Just refresh the site once it gets stuck. So then let's see view. Maybe we can zoom in a little bit more. Yeah. RNA interference, a way of silencing mRNAs. So these are the section. What is siRNA? It's more of discussing how it is evolved. Uh, Anyone? siRNAs?
Small interfering RNA.
Yeah, small interfering RNAs. And what is the difference between miRNA and siRNA? Anyone remembers the difference between miRNA and siRNA?
siRNA has more specific targets and miRNA has multiple targets.
Hmm. That's one thing. Second, during the processing from which it originates, that one main difference is there.
siRNA is double-stranded and exogenous, but
Hmm.
miRNA is endogenous.
Yeah. So siRNA, it's processed by the argonaute, one of the argonauts from double-stranded RNA, correct? And of course, the functionally, it's to shut down the translation or from the or shut down or cleaving the target RNA, just like the microRNA. But it requires perfect base pairing rather than the microRNA. So you can see, I think this will have very simple answer. See, so small interfering RNAs are 21 to 23 nucleotide double-stranded RNA molecules once incorporated into RISC complex. Okay, RNAi silencing complex RISC. They facilitate the cleavage and degradation of its recognized mRNA. Correct. So next question. Let's go. How are siRNAs made? So this one again, I think it's processed from the double-stranded. Let's go. So long double-stranded RNAs are processed by enzyme dicer. We have both the DROSA and dicer during miRNA as well as siRNA processing. Dicer is the one which processes the double-stranded RNA and incorporates into, once it gets matures, it goes into RISC complex. Correct? So then next, what are risk? Yeah, RISC. Just now we were talking about. Anyone?
RNA interference silencing complex.
Yes. So what does it do generally?
It binds to mRNA and degrades it.
Hmm. So it finds complementary sites, whether it is partial or complete base pairing. It finds and then cleaves the facilitate the cleavage of the target RNA. Correct. So basically, you can see this site is quite nice in that way. So you have RNA induced silencing complex, a combination of protein components and single-stranded RNA from the siRNA as well as microRNA, correct? It's not on the siRNA. Here is little bit problematic there. That bind to complementary mRNA and degrade it. Content, dicer, argonaute, etc. Then dicer is more of the one of the components of the RISC complex, correct, which has the RNase 3 enzyme. And you can, if you remember, there was the PAS domain which binds to nucleic acids and then targets for degradation, correct? It was there was a specific domain. If you can imagine, view last time in one of the classes we talk about the structure of the dicer complex, correct? Then next, so this one, what does the pairs domain on dicer do? This one, we know that pairs domain is the nucleic acid binding domain. That's what we discussed. So RNA binds RNA, and while RNase 3 domain cut RNA to produce two nucleotide or three nucleotide overhang. So basically It has the RNA binding function. So shRNA, what is shRNA? We talked about last time. Anyone?
Small heteronuclear RNA.
Small?
Small hetero.
Let's see.
It's
Okay.
a short hairpin RNA.
Okay. So it's
Okay.
in fact short hairpin RNA. So that's the definition of that stuff. And many times these are also used for recognition by the or recognition by one dicer processing or risk complex. So okay. So did we discuss about RNAi? Correct. We discussed about RNAi, the microRNA or siRNA mediated knockdown RNA interference. So you use RNA to interfere the function of uh, the particular RNA. Most of the time it is mRNA. Correct. So any advantage? What is the advantage? Can you anyone can come about? One of the advantages RNAi. Why does, or you imagined why the cells have evolved RNAi inside their system or in the natural system?
It allows for a transient knockdown of a particular gene. So the gene is not completely gone. You can just shut it down for a temporary amount of time.
Okay, so it's a temporal regulation is allowed with this, correct? And then second, without changing the gene.
So we discussed that the same gene sequence could give rise to multiple mRNA
Yeah.
because of the splicing difference.
Mhm.
So we could more specifically inhibit a particular protein or mRNA using RNA.
Yeah, so that is possible because transcription in many cells, or tissue type, the transcriptional termination can be different. Correct. So the 3' UTR can be different. Same gene can be there. 3' UTR can be different. Because why I'm telling you is that most of the microRNA target sites it's in the 3' UTR, but 3' UTRs can be different, and genes can be regulated in that way. Specific RNA can be regulated. Okay, isoforms can be regulated. Then anything else? Okay, let's see what. So one thing is why it has evolved so fast is that one is, of course, the advantages you have talked about is well valid, and it is a faster way to regulate gene. Okay, and technically inside it has become so popular in the research community is because the oligonucleotides can be in vitro synthesized in a uh, large number way, and it can be knocked down, or you can do genome wide or kind of screenings. All these are possible with RNAi technology. Okay, then next, let's go here. Once it comes, you just refresh. That's if you refresh, it comes back. That's what I realized. No. Let's see. Okay, this one is stuck. Let's go to the next one. The other time it was going ahead, but this time it's not going ahead. Okay, let's go to this one. Okay, so this one is all about siRNA, microRNA, or RNAi quiz. Okay, so we can start with this. Dysregulation of microRNA can lead to what kind of problem can come? MicroRNA dysregulation if it is not regulated properly. Any problem? Anyone? So basically, this answer, I'm not very much satisfied. The answer they have is it causes diseases, not always correct, but it will change the expression pattern of the tissue, or even it can change to the if the microRNA is very, very important for tissue differentiation, then the differentiation or developmental delays, all these things can happen. Correct. Basically, there will be a change in the gene expression pattern. That would be more satisfying answer to me rather than just saying disease. It's not very satisfactory. Uh, Many organisms use RNAi for what purpose? Anyone?
Regulation of protein expression and for protection against viral infection.
Hmm. For viral infection, okay, as a immunity kind of innate immunity kind of stuff. That's
Yeah,
what
you
you're
can
talking
say,
about.
yeah.
Mhm. Okay. And then to regulate gene expression, mostly is to control gene expression. Then what are two types of RNA molecule involved in RNAi? So it's very simple. What are the RNA?
siRNA.
Hmm.
uh,
siRNA and microRNAs, correct? miRNAs and siRNAs, the small interfering RNAs and microRNAs. Then let's go. The main job RNA is to do what to genes? So this is quite a redundant question. So direct gene silencing, that's what we have been talking about. But I think this is not the only answer, correct? Last time we talked about microRNAs can also give rise to, of course, it is gene silencing most of the time. It can do at the post-transcriptional level or at a transcriptional level, correct? Because it can attract chromatin modifying factors to change in the status of the chromatin organization, and it can change the transcriptional status of that particular gene or particular locus. That's what we discussed last time. Then next, describe the RNA molecule from which siRNA is derived. So we know that this one is from double-stranded RNA, correct? Then what are two ways in which long double-stranded RNA used for RNA be introduced into a cell? How can you introduce
Endocytosis.
mhm,
Viral
endocytosis?
infection.
Virus can inject the thing into
Mhm.
the cell as well.
Okay, let's maybe that is fine. Producing the soil cell itself or delivered into cells experimentally, transcribed in the nucleus microRNA. This is not very satisfactory answer to me. So, of course, you can introduce externally or endogenously, but generally is delivered into cells, correct? And from exogenously when we want to use. Of course, in the natural setting is encoded in the case of microRNAs. For siRNAs, it is generally present in the viruses, or we talk about in the repetitive elements, some of them, or transposons may have this siRNA component encoded inside. But externally, if we have to introduce it, it can be physical way, or it can be kind of endocytotic pathway that we have been talking about. So there can be different ways to introduce inside the cells. Then what is the protein to which double-stranded precursor of microRNA and siRNA bind? Precursors of microRNA and siRNA bind. Which one is that one? Double-stranded precursors. What were the complex? We talk about first after the transcription, if you remember, DROSA and what was that protein? Another one, that RNA binding protein TZCR8 or something. No? I don't remember exactly, but there are two proteins, correct? Then once it comes out, it binds to which complex? Anyone remembers? In the microRNA and siRNA processing. Yeah. So it's a dicer, correct? Dicer process it outside the in the cytoplasm. Then once it is processed by dicer, it becomes a part of the RISC complex, correct? And then silence the gene expression. So I think if we can get, you can keep on trying this quiz. This link is quite good. So it gives little bit of your answers, but this one will stop here, this question and answer session. Then we go ahead with little bit discussion on molecular cloning kind of stuff today, very fast, and then we end for today. So today what we are going to talk, most of this is the stuff, most of it is from this book, Principles and Techniques of Biochemistry and Molecular Biology, okay, by Kit Wilson and John Walker. So most of the stuff is from there. And this book, you can get a PDF copy in the PDF copy online. Okay, so you can search for this book and then you can get PDF copy of this book easily. So if anyone wants, if it is very difficult to find, then you can contact me. I can share the PDF copy, but you can just search also. It's easily available. So basically, we are going to talk about recombinant DNA a little bit. Mostly, we'll talk about how recombinant DNA. We talk about the cloning, restriction enzymes, all those things we talked in the earlier classes, correct? PCR we have talked about, then how to go for site-directed mutagenesis by inverse PCR that we have discussed in the class in the in earlier classes. So this is kind of how you can use for genetic analysis of these things, and then little bit of mm, basic cloning vector stuff, or uh, some details about the cloning vectors or vectors, and then we can end for today. So uh, so the this one is more of how do you generate kind of, if you are now it's little bit easier because you can even synthesize, or you can get the gene clones from any service provider. But when it started out, to do genetic analysis or to find the genes involved in certain process, for example, the genes which are involved in resistance to a particular small molecule, uh, like hypochlorite, for example. Hypochlorite gives lower of chloride ions and induces kind of what we call very oxidizing environment, and oxidizes many of the proteins and kills it. And E. coli and all have ways to, or genes which are involved in taking care of chloride radicals, which is coming out of hypochlorite. So if you have to find the factors involved in that kind of response, then earlier days what people used to do was you generate libraries. Okay, for example, you generate the libraries of the E. coli genomic DNA most of the time, because if you are that time people who are focusing the host is E. coli and the gene you are looking for is from the E. coli. So you just construct a genomic library and go ahead. So basically, in generating a library, what does it do is that you generate a, a collection of fragments of double-stranded DNA, okay, and that is inserted into a plasmid, okay, and that is the recombinant plasmid. What you get is a recombinant plasmid is transformed into bacteria and allow it to multiply. Once it is multiplied, you can collect the DNA and check for the phenotype that you are desired for. So how do you generate this library? For example, in the case of uh, bacteria, that E. coli we just talked about, what happens is that most of the time people generate genomic DNA library. But if you want to generate a library of genes controlling or responding responsible for a certain phenotype in eukaryotes. As you know, in eukaryotes, you have introns in the genes, and the gene size, including both the exons and introns, is going to be very, very big. So in that case, cloning, for example, one of the genes I'm interested is like uh, ABCA1. Okay, ABCA1 has 2,700 amino acids. So that means if you look at the coding triplet, is going to be around how much? 7 kb. And 7 kb of the gene, which is fragmented and getting it in one go in the uh, bacterial or eukaryotic expression vector, it requires to get the direct sequence of the mRNA. Okay, so that means you need to have you have to generate a library which contains only exons. Okay, these exons containing libraries can be obtained from the mRNA collection by something called cDNA library. So you can have two types of libraries. One is genomic library or genome library or cDNA library. The difference here is that in the case of genome library, you collect the genomic DNA and then generate a collection of double-stranded DNA fragments from the genomic DNA, and that is cloned into a plasmid, and you can look for the presence of the phenotype you're looking at. And generally it is very useful for something like in bacterial genes, or if you're looking for just presence of a uh, uh, certain element, repetitive elements. For example, if you want to look for repetitive elements sequence, whether it is present in one organism, is also present in another related organism. In that case, you clone genomic DNA library and probe with certain probes. It can be a DNA probe or RNA probe, or you can do PCR and then identify those fragments. So genomic library is just getting a collection of fragments of genome and clone into a cloning vector. In the case of cDNA, complementary DNA library, okay, the difference here is that the mRNA, it starts with mRNA in this case. You don't start with genomic DNA. You start with mRNA. From that mRNA, you get the complementary DNA. What do you mean by complementary DNA? The DNA which you synthesize by putting a primer which is complementary to mRNA, so that you can get a complementary strand to the mRNA you are getting at. So you get complementary DNA. That's why it's called cDNA. Once you get this cDNA, the first single strand you get by extending the primer complementary to mRNA, you get a complementary DNA. But To clone as a library, you need to synthesize the DNA, which is complementary to the cDNA again. So it requires the synthesis of both the cDNA strand as well as complementary to it. So then finally you get a double-stranded DNA which can be cloned into a certain vector of your choice. If it is for bacteria, you will have a bacterial promoter followed by cloning of this double-stranded DNA so that you can get the protein expressed in bacteria. But if you want to express in eukaryotes, then you need to have a eukaryotic promoter and other elements we can discuss later in a next class. What all elements are going to be there in this expression vector is. But for time being, we need to, the difference here is that in the genomic library, you start with a genomic DNA or genome of that uh, particular organism of interest. In the case of cDNA, you start with mRNA as the starting material. So for genomic DNA library, you can do many a times, very popular is the restriction enzyme digestion. Okay, restriction enzyme digestion, for example, that we discussed in the earlier classes, something like EcoR1 sites, which has a recognition site of GAADTC, that is 6 base pair recognition site. It is likely to happen once in 4096 nucleotides, correct? Because 1 by 4 into 1 by 4 into 1 by 4, that's 6 times. Okay, so that would give you 1 out of 4096 nucleotides. So that kind of if you take a genome which is supposedly balanced 80 or 50% GC content, you are likely to have one ECOR1 sites in every around 4000 nucleotides. So if you use ECOR1 or some other enzymes which recognize the 6 base pairs, you can generate a kind of fragment which is around 5 to 10 kb depending on the organism. So you can digest weight, restriction enzymes, and generate fragments. Okay, for example, here if you look at what people generally do is treat with the enzyme, but you treat for a limited time, that so that the digestion is not complete. So that is called partial digestion with restriction enzymes. Why you are doing this partial digestion? Because you get with the same restriction enzyme, for example here E, suppose we think that is equal 1, it can generate all these fragments by complete digestion or partial digestion. 2 of the fragments can be joined together, or 3 or 4 can be joined together. There is a population of fragments which you can clone. But if you go for complete digestion, you are going to get, for example, this genome will give you only 1, 2, 3, 4, and 5 fragments only. But if you do partial digestion, you can get 5, then 6, 7, 8, 9, 10, 11, 12, 13, like different numbers. Like you're, instead of 5, you're going to have multiples of that in the DNA fragment. So partial digestion is very, very much well utilized in many of the cases in genomic DNA liberation. You digest with certain restriction enzymes and then try to put in a cloning vector. Then recently, there are techniques. What people do is, instead of treating with restriction enzymes, you do a physical treatment. For example, sonication. Okay, by sonication, what do you mean? You put a sound, sonic waves, sound vibration, and then allow the cleavage or the fragmentation of the DNA, and you can get, you run on a gel and select the size you want for 5 kb or 10 kb long size. You cut out the gel and put that in a cloning vector. That can also be done. Okay, so you can do by restriction enzyme digestion, or by physical fragmentation of the genomic DNA to get library of fragments, which once it is cloned into a vector becomes a genomic library. In the case of cDNA library, what you do is you isolate the mRNA, and as you all know, the eukaryotic mRNA has poly A tail. So many a times people utilize poly T primers. Okay, you put an oligonucleotide of stretch of T's so that it can bind to this poly A and extend so that you get the first cDNA strand. Okay, first strand of, sorry, this light one, first strand of DNA is synthesized. Okay, now by doing this, there are different ways. You can use poly DT primer or oligo DT primer, or you can utilize random primers. Random primers is like 20 base pair long oligonucleotide. You randomize during the synthesis. What you do is, instead of specifying particular base, you say any nucleotide can be incorporated at the first position, second position, third position, so you can get a library of oligos. Those are called random primers. So these random primers, when you throw in, what will happen? It can bind to the mRNA at different positions, and it can extend the cDNA library. This generates the first strand of DNA, which is complementary to the mRNA. That's called cDNA mRNA hybrid. Okay. Now, to make this uh, single-stranded cDNA into a double-stranded cDNA, how is it go about? There are different ways to convert this single-strand cDNA into double-stranded cDNA. The one of the well-known mechanism is that you use the oligo DT primer so that you get the first cDNA. But cDNA, the first strand is synthesized. Then what happens is, many a times the reason for this is not yet known. Maybe it's because of the sequences in the 5 prime of most of the eukaryotes. It forms a hairpin structure. Okay, so what does it mean? The hairpin structure means you have complementary region to 3 prime. Now this hanging hairpin end, 3 prime end, can be utilized as a primer for the synthesis of second strand. So this 3 prime end can be extended by DNA polymerase to synthesize this kind of DNA. Okay, so double stranded DNA, and it's kind of a hairpin loop. It's a kind of loop. But what happens is that these long DNA, single DNA, can be converted into double stranded DNA because the loop structure has a hanging single stranded region, single stranded DNA region. So this single stranded DNA can be clipped with S1 nucleus. So S1 nucleus is a DNA nucleus which cleaves single stranded DNA but doesn't touch the double stranded DNA. So S1 nucleus digestion will remove this linking single stranded loop. So then it converts into double stranded cDNA. So this double stranded cDNA can be utilized for cloning to generate cDNA library. Okay, so next, what other people do, another strategy is, there are, of course, you can see here the hairpin loop formation is not so efficient, and it's not very good approach to generate whole cDNA library for an organism or particular cell type, because not every mRNA after the first strand synthesis is going to form this hairpin loop very efficiently in all the mRNAs. So some mRNAs will be amplified much better. Some mRNAs are not at all amplified. Some are just amplified very little. So there is a discrepancy there. There is a discrimination from that side. So what people do, another way is you do after the first strand DNA synthesis, you treat with RNase H. Okay, and RNase H, what it will do, it will cleave the RNA into fragments. As you can see here, these fragments can be complementary to this first strand cDNA, so that the RNA fragments can serve as primer. It's very much like the random primer, correct? The random primer we discussed. So it's just like the random primer. It extends and synthesizes the double-stranded cDNA. But you can see here, again, there is a problem here. All across the length of the cDNA first strand, you have multiple primers, and not all of them is going to give the full length protein coding sequence. So most of them will be a truncated double-stranded cDNA. For example, in this picture, only this primer is going to give you a full-length coding double-stranded cDNA. So there will be a lot of truncated double-stranded DNA, cDNA. So there is a disadvantage, but depending upon your requirement, this approach can also be very useful, because if you are looking at just the expression or generating a probe for something, maybe it's good enough, correct? So the other way many people utilize is, what you do is you try to add extra sequences, poly Cs or poly As, so that you can amplify all the CNN in a full length in a better represented way. So in this case, what you're doing, after the first cDNA strand is synthesized, after the first round of primer extension, oligo-DT, then this DNA is treated with DCTPs, or most of the time DCTPs are utilized in a commercial setting. So DCTPs are added using terminal deoxynucleotidyl transferase. What it does is that this enzyme will add few cytosine bases, okay, Cs. So this Cs now is extended at 3 prime. Now if you add a primer wherein G is the poly G as a primer, then this poly C will base pair with poly G, and you can synthesize the second strand and you get a full length cDNA. So this approach looks quite good, and it represents better for generating cDNA library for your gene expression purposes. Okay, then after this, what you do now, once you get the cDNA library or genomic DNA fragments, what you can do is either you can blend and edit it depending on the purpose you are getting, and you can generate cohesive ends like this. In this case, you put in the primer that in this purple segment, you put a BAM-H1 sequence in here. Then what you can do is you can digest those cDNAs with BAM-H1 and clone into the BAM-H1 digested cloning vector, and you can get clones of this. So that's how you can generate genomic library or cDNA library for your studies. Now, if you are looking for utilization of these libraries for certain purpose, this example can be applied directly for prokaryotes. It was utilized even for eukaryotes also. In the yeast field and all, people have utilized these. What people do is you generate genomic DNA library or cDNA library, depending on the organism you want to do. Now you have a library. Suppose because of a mutation in a gene, the organism becomes temperature sensitive. What do we mean by that? Because of the mutation, the protein or the locus becomes inactive, and the organism stops to grow. You can see a lot of TS mutants in the case of E. coli. You will have temperature sensitive mutants of Saccharomyces cerevisiae. Now you want to, if you got that mutation because of treatment with chemicals which introduce random mutations. For example, the treatment with mutagenizing chemicals, you introduce mutations and get a temperature sensitive mutant. Now you want to find, so this was very common earlier. So now you want to find what is the gene responsible for it. What you do is you generate genomic DNA library or cDNA library and transform that into that TS, temperature sensitive strain of E. coli or the eukaryotic like cerevisiae. Then you shift up to non-permissive temperature. What do you mean by non-permissive temperature? The temperature at which the temperature sensitive mutant stops to grow, but the wild type strain still grows. That is the non-permissive temperature. So after transforming with the library cDNA or genomic library, if you want to find out, you transform it and grow up at higher temperature, that is a non-permissive temperature. What you will do is only the genes which is complemented. What do you mean by complemented? The defective protein is supported by a copy of wild type in the plasmid. So that is the complementation, right? It is complemented and allow the organism to grow at non-permissive temperature. Now, once the organism grows, what will do? You will get multiple copies of the gene. Okay, so these multiple copies of the gene you can use for finding the gene of interest. What you would do? You would sequence these fragment, the fragment you have cloned, and find out what gene is there in this plasmid to know the gene responsible for that temperature sensitive phenotype. So you can find genes. These libraries are very useful to find genes responsible for a particular phenotype. Okay, so next, so now in the case of E. coli or some of the vectors which are very much used for this kind of gene library preparation, and some of the plasmids are something like M13. Now it is again becoming popular. Earlier days it was very popular because of the fact that it's a single strand DNA virus. Okay, M13 viruses were pretty much used to generate mutations that we discussed earlier also, correct? And then we do use plasmids, then we use viruses, and combination of virus and plasmid, those have cos site in this site called cosmids are also used. These cosmids are combination of bacteriophage elements and plasmid elements together. Okay, the why you evolve all these things is that you can see the insert range, how big your DNA fragment can be. Okay, so depending on that, these different systems can be utilized. Then later on, to increase the insert size, you do get bacterial artificial chromosomes. Then you have yaks, yeast artificial chromosomes. Even human artificial chromosomes have been uh, utilized recently. So now, what is the importance of these things is that the artificial chromosomes, it can take insert size which are much bigger than the plasmids, generally 1 to 5 kV. Very efficiently you can treat. Of course you can see much bigger inserts as well, but generally the insert size is not so big. Okay, so these are different vectors that can be utilized for generating libraries to study certain phenomenon you are looking at. So one of the first E. coli vectors, plasmids, which has been used is the one of the vectors called PBR322. This first generation plasmids. Okay, so in this kind of plasmid, this is a cloning vector. What do you mean by cloning vector? It doesn't facilitate any fragment or the fragment to be expressed when you put the clones inside. So basically, you need to have a origin of replication that we discussed earlier also. Origin of replication, and uh, what is the time now? Origin of replication, then you have to have a selectable marker and you have to have cloning site. In this vector, what is the importance of this second marker here, tetracycline, that can be used to isolate or to find out vectors wherein inserts are there or not. So for example, if you are cloning something in BAMH1 site, what you can do is that because insert has come, the translation or this ORF can be disrupted, and the genes, the insert or the plasmid which have insert at BAMH1 will no longer be resistant to tetracycline treatment. So you can select those vectors which are not growing on tetracycline, and you are sure that there is some insert at the BAMH1 site. So basically, in a vector, the basic premise of utilization is origin of replication is a must. Okay, and for our convenience, you will need to have a selectable marker. Okay, selectable marker, it can be ampicillin or whatever you can select for. Okay, ampicillin, and something you need to have a cloning site or multiple cloning site that we talk about. So among the E. coli vectors, what is important here is that the origin of replications can be different in many of the vectors. For example, we talked about PBR322. The origin of replication which was used in PBR22 is called PBR322 ORI, and these are used in many of the protein expression vectors like PET system, PZAC system. Okay, PET system is very much utilized. PZAC is also very much utilized for protein expression in E. coli. Okay, so if you look at it, you have different types of origin of replication. PBR322, one is PBM1, COLE1, R6K, P15A, correct? PSE101, COLE1, bug, and F1. So there are different types of origin of replication. But if you look here, it can be classified into three groups: Group A, Group B, and Group C. What does it mean is that it requires two things. One is this origin of replication controls the number of molecules. If you put PUC, okay, with origin of replication PPM1, gives rise to lots of copies of the plasmid inside E. coli. That's called the relaxed state. It keeps on dividing, and the number is much more inside the cell. These are relaxed plasmid. You can get multiple copies. See here, 500 to 700. If you put into PUC, you will get 500 to 700 copies of the gene you are looking at. Okay, so and stringent means the number of copies, number of plasmid is very less inside the system, because the replication of the plasmid is closely, very much linked to the division of the host cell. Okay, so again, the compatibility group A, B, C. What does it mean is that when you put inside E. coli, the A compatibility plasmids cannot be put with another A plasmid. Okay, it can be put only with B or C. Likewise, B class of plasmids cannot be put with another B class of plasmids, because they are incompatible, and the cell tries to maintain only one plasmid which has the same group. Like A group will maintain only one, C will be maintain only one, B will be maintain only one. So if you want to utilize three different types of plasmids in your screening in any of the experiment, you have to put one A type of plasmid, another B type, and another C type. Then only you will be able to put three plasmid in the same E. coli cells. Otherwise, you will never be able to maintain three types of plasmids. Okay, so this is what is. So whatever we have been discussing, that the tetracycline stuff, what we were talking about earlier, yeah. Okay, so we'll take another 2 minutes. We'll finish with this slide for today. So what that tetracycline gene, when you disrupt it, you said that that is called disrupted cloning. So what you do is you allow the plasmid containing cells to be selected with the selectable marker, that is ampicillin in our case of example. So these ampicillin resistant colonies make sure that those cells which have the plasmid is grown. So it's selected for the plasmid. Now you want to check which cells have inserts or not. That you can do by replica plating. What do you mean by is that you put a paper and then put, it's just like the printing press, you press it down so that you can get a replica of the colonies on the plate. You press the paper, take it out, and put into a fresh plate which contains tetracycline. So when you put in press again, you get a carbon copy or replica of the colonies on the selectable marker. Now, once it is replica plated, what you can do is you let it to grow, incubate it. So in this case, some of the colonies, for example, these kind of colonies will grow up. That means there is no insert in those colonies. And another type of colonies, something like this one, which doesn't grow up in tetracycline. That means they become sensitive to tetracycline. So during the cloning, the tetracycline has been disrupted and unable to grow on tetracycline selectable marker. So these colony and this colony has inserts. These can be selected and put into your what can be in your collection of cDNA or genomic library. There are other types of disruptive cloning. For example, many people have used, instead of tetracycline, you use a toxin uh, ORF, okay, toxin. For example, very, very popular one is CCDB, okay, CCDB. Which, if you have CCDB, it will kill, because it is a toxin, it will kill the cells. So what you do is you clone it and try to disrupt that CCDB open reading frame. Once it is inserted, what will happen? That CCDB ORF is disrupted, so no toxins are produced, and it will be growing on the cells. So basically, that will allow single selection. In this case, ampicillin and CCDB disruption. These kind of colonies where there is no insert will not grow when you use this kind of selection. So these are called negative selection markers. If you use negative selection markers when you have the gene, it doesn't allow to grow. When you disrupt it, it allows to grow. So you can use those kind of toxin genes for disruptive cloning, so that whatever is growing up on the plate has insert in those colonies. Okay. So far, this is the generation of cDNA library, and then there are different ways to select for the clones being there. Okay, I think we can stop here for today, and we will meet in the uh, next class. And I think I'll try to wind up by next week. Okay, let's wind up with another 2 classes in the next week and then finish it. Okay, so I'll stop here for today. Yeah, stop.